Okay, so welcome everyone. Thank you for joining us this evening for a very exciting webinar, developing a custom test menu for urine drug testing. I'm Vasil Kapitan. I'm with marketing team at Lighthouse Lab Services, and we have with us Sarah Harris, our presenter, and also uh, Robin Hoyle. She's executive director for Pain Societies, our partner, and I would like to hand things off to Robin to make a quick intro about the series, about herself, and then we're going to get to the intro for Sarah and presentation.
Well, hello, everybody. Thank you so much. We're very excited to participate and to partner with Lighthouse Lab Services on this series. Well, this is our third of four. It's been a lot of fun. The last two topics were outstanding, and we do have those videos available for anybody that would like to go back, review them, or share them with your staff at the office over lunch on a Tuesday. They're all available at Lighthouse Lab Services website, and we'll make sure that we send you that link. The one other item of housekeeping I have for you tonight is regarding the CME. You do get AMA PRA Category 1 credit hour for tonight, but please don't claim your CME till the end of the full series because you can only claim it once. And we do have one more fantastic talk after this one in this series. It will complete on December 13, 7 p.m., all about coding and billing, which is also very important. So we hope you join us for that one as well. And I will hand it back so we can get started and hear this excellent presentation. Thank you so much for the support of this. We appreciate it.
Thank you, Robin. And now we're going to go to introduce Sarah Harris. Sarah Harris is amazing at what she does, and she has a lot of expertise. She earned her master's in chemistry from University of Notre Dame and her PhD from public health from University of South Carolina. She's also certified toxicological chemist with the National Registry of Certified Chemists and has nearly a decade of extensive experience in method development, optimization, validation across a wide array of laboratory disciplines and instrumentation. And also in 2016, Sarah, she became our Chief Technical Officer for Lighthouse Lab Services. She's been great at it. This role involves building hundreds of laboratories, many of them specializing in toxicology, and with clients across the nation, collectively processing more than 100,000 samples per month.
Wow.
So thank
Well,
you, Sarah, for everything.
thank you for that warm welcome. I really appreciate it. Very excited to be here. Good evening, everybody, on this Wednesday evening. Appreciate your time. Thank you for taking the time to attend today's webinar. As Vasil mentioned, I received my master's in chemistry at the University of Notre Dame. I focus mostly on metabolomics and specifically LCMS methodology. I then moved on to complete my PhD, and that focus was in the opioid epidemic. So that's kind of where those two worlds kind of came together there. Since then, I've worked in a lot of toxicology labs, creating custom test methods and validating those for laboratory developed tests. So we're excited to be talking about that today, and we'll get right into it. If I can get the slide to move. Okay, there we go. So whether you're creating a custom test menu for your in-house lab or curating a test panel for sendouts, selecting from the ever-growing list of medications and drugs of abuse that are available to test can be challenging. Also, a question I get very often is, when does it make sense to add additional targets to a current test menu? And is there a time when perhaps it doesn't make sense to keep something on a test menu any longer? So those are going to be a few of the topics that we're going to discuss today. So first, we're going to be talking about targeting that test panel to fit the patient population. Just a refresher on the types of testing, because really you'll need not one, but two different panels, one for your screen and one for your confirmation. Working through both that presumptive and definitive test menu, and then proper utilization for subpanels. You'll have your main panel, and we'll talk through that, but then you'll often have several subpanels for different types of testing, different patient outcomes. And finally, compliance with that LDT regulatory requirements. If you will be validating these test venues in an in-house lab, you'll want to ensure that you are staying compliant with that LDT regulatory requirements. So one, we're going to talk through targeting the test panel to fit that patient population. I want to do a little bit of stage setting here, and I first wanted to touch on what I'm referring to when I talk about test panel. So first, we're going to talk about that full test panel. This is the suite of drug classes that is either validated on your clinical equipment in your lab, or is all that suite of testing that you're able to select from on that test requisition form if you're currently sending those tests out. So that test panel is going to be it's going to encompass all those different orderable tests, both on that EIA side or your screening side, and then that LCMS typically, possibly GCMS if you're sending out on that definitive side. And then we're going to move into those subpanels. And this is going to be this difference between different patient and testing types, often referred to as profiles. And these are built from that larger panel and are used for different medical necessity scenarios. This could be your intake test panel, a randomized subpanel, high versus low risk, et cetera. And we can talk through those a little bit more. But kind of just wanted to set that stage when we're talking about that full test panel or what's going to be validated on that clinical equipment versus subpanels that physician is going to be curating for different scenarios. So first, evaluating that patient population. That's the first thing we always look at when we're recommending a test panel or when we're designing a method that we're going to validate for that clinical equipment. Are the patients being treated for chronic pain in a pain management setting? Or are they in a substance use treatment center or going through MAT? Those test panels might be different depending on that patient population. Also, regionality. We like to do an assessment of the region and what drugs of abuse are most common in that area. It can be different between state and also rural versus urban. So those are some things that we might be looking at when we're kind of curating that test menu. Also, state laws come into effect. We've had several practices choose to exclude THC recently based on changing laws in their area. They found that as a practice, they've decided that they were just not going to include that and have that physician having to make those calls based on that changing law. And then finally, polypharmacy with medications that are not considered drugs of abuse, but that may interact with commonly prescribed medications in the practice. Medications that might potentiate drug effects, making an overdose more likely in that setting, or medications that might compete with drug interactions and metabolism. Those might be ones that are looked at to include, even though they're not typically thought of as drugs of abuse on that test panel. So here's although there's no standard test panel, here's some commonly best practices that have been kind of agreed on in the literature. First of all, testing should only be used in conjunction with self-report and clinical assessment, and this is really to obtain that full clinical picture. But as for the panels and the testing, first, that initial presumptive toxicology panel, most experts would recommend that the program would include the most common substances in the local community, any public health concern substance, which example would be fentanyl, most commonly xylazine, which I think y'all talked about in your last webinar, and any drugs of abuse that may be coming onto the market. Any substances of particular clinical concern for a patient. One that's commonly talked about is the Trinity, which is carisoprodol with an opioid and a benzo. Carisoprodol might not be something that's prescribed in the practice or commonly on its own a drug of abuse, but perhaps when it's prescribed with other medications might be of concern. And then confirming the presence of therapeutic medications. This would be buprenorphine, prescribed benzodiazepines, methadone, things of that nature. But really, the test menu all comes down to what do the providers need to make an informed clinical decision. That's really what it comes down to. What is going to be on that report that is going to offer the most information to the provider and offer the best outcomes for the patient. So number one, when that intake and that new patient, making sure that all of that information is there upon intake when you're forming that new relationship. So identifying any undisclosed prescriptions or illicit drug use from day one. It might not be intentionally illicit. Those might not be the ones of concern in your practice. It may be a medication from another prescriber that the patient didn't even think that they needed to mention, that might have an interaction with something that might be prescribed in your practice. insight to behavior ahead of treatment-related decisions. So you can alter that patient's current risk assessment. The patient panel should really go hand in hand with those risk assessments that are being documented in the patient's chart. Chronic pain treatment practices often require acceptable drug screens prior to prescribing scheduled medications. So ensuring that there's enough information on that initial intake report to be for that safe prescribing of opioids. And then substance use clinics document these for treatment progress and watching for any type of regression. Finally, medication management for established patients. Ongoing random drug tests can ensure that compliance with a treatment plan and dosing regimen is consistent throughout that provider-patient lifetime. So we're going to talk just a little bit of a refresher on types of testing and the types of panels that need to be curated. So we have both the screen and the confirmation. First, on the top here, we're talking about the screen. That's that preliminary testing. It's often very quick. It can come back with minutes to hours. It can be a benchtop or maybe a larger floor model, but it's going to be looking for the drug classes, not the individual specific drugs. So we're going to be looking for the opioid class or the benzodiazepine class, not the individual medications. So they can be the results can be available faster, possibly even while that patient's in the office. And they're a lot of times useful in ruling out negative results, but they often lack the detailed information and the quantitative information that secondary confirmation can tell you. So the definitive or confirmation is most often run on that LCMS, sometimes GCMS, and they produce definitive or confirmation results. That's going to not only tell you that this screen was correct or perhaps was not correct, and it's going to tell you the amount in the urine or oral fluid if you're using a different medication, and the exact drug in that drug class that is present. Results are a little bit slower. They can be days or weeks sometimes if you're sending out to a lab, one of the larger reference labs, but those will be the most accurate results, and most testing protocols do require some type of confirmation of those initial preliminary results. Things to think about when you're doing curating your test panel would be the matrix. Today we're talking about urine, but if you do also test for oral fluid or another matrix in your practice, that test menu will likely not be the same. Urine, your detection windows are 2 to 5 days, and you're often looking for metabolites long past when the parent is able to be detected. That parent will have metabolized throughout the body. Oral fluid, on the other hand, a lot of those metabolites wouldn't be applicable. They wouldn't be in that oral fluid. So you want to make sure that when you're developing your test panel, you're not including metabolites or analytes that you wouldn't expect to see in that particular matrix. All right, so now we'll really get into it with a little bit of examples of presumptive and definitive test menus. So again, we talked about this. There's no universal standard, but a good place to start. Policies would include reasons for testing, how results will inform treatment, a general approach to frequency, circumstances for ordering the definitive testing, and how should providers decide which substances would be included in that testing. That's what we're going to focus on next. So what to pick? On the left here would be an example if you were starting a lab in your office, or you just got a new analyzer and that vendor gives you a list of here's all the options that we're able to run on this EA analyzer. You select that they're kind of like this is your options to choose from. So you can see them all on the left. There's often a lot of overlap. For instance, there's an amphetamine only screen. There's an amphetamine plus ecstasy screen. There's an ethyl alcohol screen versus an ETG screen. There's really a lot of different options. And this is just one vendor. Many other vendors have additional more select tests where they're newer. Tramadol has a screen now. Gabapentin has a screen now. There's a lot of additional medications that are coming on the market that have a screen. So things to consider when you're looking at this, this test panel. Which results are going to go to definitive testing and what positive results are going to go to definitive testing. Cost, including every single screen may be more costly than the reimbursement itself. So that's something to consider, both cost for the clinic and cost for the patient. Positivity rates. If you run a screen for a whole year and never have a positive, it might not be a screen that you want to continue running. And then specificity. You want to make sure that that screen that you're looking at is accurate and giving the information that you need. If 50% of the time that screen is not giving you an accurate result because it's cross-reacting with another medication, that might not be one that you want to select. So let's get into a few examples. For instance, a question I've gotten when we're looking at these test menus and kind of determining what to validate on a piece of clinical equipment. Let's start with the opioid group of drugs. There's a heroin, a hydrocodone, an opiate, and an oxycodone screen. Well, do we need all four? Do we need one? What do we need? So things to consider. Is more information being gained from testing all four? And does this change the number of definitive tests needed? So for instance, if you prescribe hydrocodone often at your practice, is that going to be tested for by the opiate or picked up by the opiate screen in this case? That's when we would look into the product inserts and look at that cross-reactivity. If every single time both of those are going to come out positive, it's most likely not effective to include both of those in your test panel. You probably only want to include one. Whereas it might be a good idea to include the opioid and the oxycodone screen because those have different cross-reactivity rates. And then looking at what you're going to reflex to definitive. If, for instance, every time you have an opioid positive screen, you're going to reflex that into that differentiative, the definitive panel, and that panel includes heroin, it might not be needed to also have that heroin screen because you're going to be looking at it on the definitive. And the primary metabolite of heroin is morphine. So it would also be picked up by that opiate screen. So those are the things that we look at when we're kind of determining a test menu. That number one is looking at that cross-reactivity, actually going into what the manufacturer lists as cross-reactivity. They'll always list a concentration that these will be positive for all the different individual drugs that they've tested. And it will almost always include the suite seen here. So that's going to be really important to make sure that you're not getting duplicate information by running a screen that's very similar and then reflexing it to the definitive. Another example that comes up, for instance, is cotinine and ETG and some that may not be considered medication drugs of abuse. Things to consider in this case. Are these outlined in your patient prescribing agreement? And do providers have a clear understanding of what to do with positive results? More results can often lead to confusion if clear expectations are not set up front. For instance, is there a medical decision being made from a cotinine positive? If there is in your practice and there's a full understanding of what to do with that result, both on the provider and the patient side, and that's an effective screen that you might want to add. However, if there's no change to clinical decision-making from the screen, this might not be a medically necessary screen to have included in your test panel. So if we thought there was a lot of options to choose with the screen, there's even more with definitive testing. Screens are often FDA cleared and have a very precise testing methodology. You have to follow the IFU. They're FDA cleared, so they include all of the kits and they have very clear cutoffs. Whereas definitive test panels for urine toxicology are all laboratory developed tests. There's no current FDA cleared methodology. And advancements in the technology used for these testing, the LCMS instrumentation, has come a long way in the last decade. You can run more analytes than ever on a single method. You can get lower cutoffs than ever. So it can make deciding what to include even more challenging because the options become limitless. So here's a method that I just pulled from literature. You don't have to read all the different compounds, but there's 177 different compounds, medications that this particular method is looking at. That's a lot of different compounds to choose from. From a validation perspective, to try to meet all of the regulatory requirements to get these through accuracy and precision, and we'll talk about that in a little bit. And then B, there's a lot of justification on the provider to ensure that these are medically necessary tests. So while you're creating your test menu, again, more isn't always better, making sure that the tests that you select for your test panel make sense to your patient population and your prescribing. That's what's going to be an effective test panel. And then having positive results that are actionable is also very important, that if there's a positive result that's unexpected, that there's an actionable item that can happen for clinical relevance. So when to add to your test menu. Emerging drugs of abuse. If there's a notification from the CDC or FDA of an emerging drugs of abuse, that's one that may be a very straightforward add to your test menu. If there's a medication that is going to change treatment, that may be one that you'd want to add. And then increased risk for overdose. As more literature comes out on polypharmacy and things that may interact with opioids, those might be literature-based approaches to change a test panel and have proper documentation for utilization. But it's not always just about adding to your test menu. It's also about removing. Change in abuse rates. One that I'm going to highlight here and a little bit later in some of our real world examples are synthetic cannabinoids and bath salts. We've seen a major drop off in certain populations where test rates might have been 5 to 10% of the population before, and a practice hasn't seen a positive in a year. And so that might be something that the synthetic cannabinoids kind of shifted as a legalization of marijuana has happened in a lot of states. And then bath salts, again, other drugs of abuse have kind of taken the spot of some of these previously high-risk drugs of abuse. So assessing that test panel, both for additions and the removal of some items, is critically important when you're looking at it from an overall perspective. Okay, so we've talked about the large test menu is what can the instrument validate? And we know we can validate a good bit on the definitive side. But after we have everything we can order, it's breaking that down into subpanels. And that's what we're going to talk about next. So the question is unique because often these subpanels are going to be changed depending on the type of testing that you're doing. An intake admission test might have a larger subpanel than a random interval test throughout the treatment. And so crafting these subpanels for very specific scenarios and creating a policy in your practice of knowing when to use each subpanel is definitely going to help if there's any type of utilization audit and medical necessity for reimbursement. And I know you're going to be talking about that in your next webinar, really the billing side of things, but I'm just going to touch on it a little bit here because it plays into selecting that test menu. So if you haven't seen these before, these are the primary, definitive, and presumptive CPT codes for high complexity testing. And they're grouped into these drug classes. So you start with your G0480, and that's going to be your smallest panel, 1 to 7 drug classes, and you move up through that into 22 or more. There's also a presumptive code, and that's regardless of how many tests in that presumptive, that can be any number, and that includes specimen validity. So as you're crafting your subpanels, it may be important to look at also how the payers are kind of identifying these categories as well. So perhaps your intake, when you first see a patient, that might be a larger test panel that has 22 or more. And as the patient kind of goes through treatment and is adhering to treatment and doesn't have any high-risk behaviors, you're creating additional subpanels that fall into these categories. And this will help during those audits to make sure you're not having overutilization of testing. So as a summary, when you're looking at creating these subpanels, include drug classes and specific drugs and metabolism that are most often used in patient population. Obviously prescribed medication, whether it's an opioid pain prescription or your MAT prescriptions, those would be in perhaps your core panel. And then they should also use definitive toxicology to confirm positive presumptive results or negative expected results. And that can be something that you might want to craft in that second tier of your panel, utilizing that reflexive procedure to confirm those results. Avoid including every possible medication. This streamlines reports for the providers, a lot of unnecessary information if it's A, not clinically relevant, or B, not relevant to your prescribing. And then it will also reduce that billing scrutiny for unnecessary testing. And finally, on that definitive, I did want to touch on that most often definitive will have a quantitative substance level. However, they aren't always clinically meaningful. You should proceed with caution on interpreting those quantitative values because they can vary drastically based on hydration or creatinine and blast dosage. If there is any type of therapeutic monitoring that needs to be done for the patient, that really should be done in blood, which we haven't talked through today, but is really, if you're looking for that quantitative value to alter prescribing dosing, that's when we should move into that therapeutic drug monitoring, which has a different set of test panels as well. And then there was just a few more examples of the subpanel options. There's some conservative approaches which could be employed where you only confirm based on that positive screen, and that's that reflex that I mentioned previously. And then there can be static panels that are also crafted. So those are the two main approaches. One would be that reflex option where only positive or unexpected negatives are then sent to the definitive. And that can often be done automatically through an LIS, of course, with the provider's order, but that workflow can be set up. So it's more of an automated solution. And then there's the other option where you're creating those subpanels based on that type of visit or risk behavior. So those are the two main approaches that are kind of defined when we're looking at those custom test panels. So if you're looking to do this in your office or at your local laboratory that maybe you send to that you're working with other offices to try to bring up some additional test panels, there are some LDT regulatory requirements that we should be made aware of and will help kind of crafting not only the test panel, but then cutoffs are important as well when you're talking through making sure effective monitoring. So one, what is an LDT? Again, like I said, these are non-FDA cleared or FDA approved tests. All urine toxicology tests that are done on LCMS are going to be laboratory developed tests. They still do go through rigorous scrutiny. The CLIA will oversee these type of tests. There's also additional regulatory bodies that your labs might utilize, such as COLA and CAP, but everyone must meet the minimum CLIA requirements. You can find more information here. I'm sure when the slides go up, you can take a look, but there are set requirements for an LDT that any lab needs to adhere to. Here's an overview of some of the tests that would be required during that validation. And that's both if you're adding a test, you would still have to go through all of these, or if you're developing an entirely new panel. So first, you're gonna have to look at your accuracy. Does the test result in the concentration in the expected range? We're looking at the bow and arrow, are you getting it in the center? Also precision, if you test the same sample over and over again, are you going to be getting the same result consistently over time? Both of those have to be looked at over a period of days during the validation of these analytes. Secondly, matrix effects. This is one of a large one when you're looking at new analytes, or if you're doing something that's relatively novel or has just come to market. Recently, xylazine was an example where a lot of labs had to bring this up and there wasn't a lot of testing previously. So matrix effects needed to be rigorously studied because it can lead to suppression or even interferences, which is the next one on the list here, that might interact from the urine or other medications that are being tested for. So looking at both of those. And then carryover is of a large amount in one patient and the very next patient going to carry over into that and cause a false positive in the next patient. There are some pretty high standards that needs to meet when you're looking through these laboratory developed tests. And then finally, a patient correlation. You always have to compare some patients in your population to a previously validated CLIA method to confirm accuracy of your particular method. And something else I'll note is when you're defining the method and the validation, the instrument may be able to go extremely low, very low limits. Like I said, the technology is improving dramatically over the past few years, but that's not always best for clinical reporting. Differences, for instance, in ETG, even morphine, you'll see different cutoffs based on that regulatory. SAMHSA is going to have higher cutoffs for their methodology because they're DOT testing. For instance, for their opioids, they raised them to where there was concerns over poppy and if there could be some positives from some of those substances. However, in an abuse setting or a pain management setting, the cutoffs might want to be a little bit lower to make sure that you're able to detect that ongoing prescribing. So making sure that you're looking at the cutoffs, not only what the instrument can perform but also what makes clinical sense is important as part of that laboratory developed test validation. So lastly, I'm just going to go through some real examples of test menu additions and subtractions that I've had questions about or worked with practices on over the last year. So these are all real that we kind of worked through or talked through. Xylazine obviously was one that we've worked on, but I won't include that since you talked through that with Eric on the last, Dr. Corte on the last webinar. But that is one that came up quite often and we have added to several labs around the country. So this was one that we had where ketamine, a practice had added a provider who was prescribing ketamine as an alternative pain therapy. Ketamine was not in their panel, but it was part of their drug testing program that all prescribed medications should be tested for to adherence. So this was something that the practice as a whole knew they needed to add. They wanted it to match their prescribing, their test program. And so this was something that the physician, the providers did decide to add to their test menu, both for their send outs and they did have an in-house lab. So we went through the full LDT validation, ensured that it met all CLIA requirements, and they were able to add this to their test menu. Other physicians also after that decided to add this as part of their profile, just to keep it consistent across providers. We had another addition. It was an MAT center. The practice had a no alcohol clause in their patient agreements, and several patients appeared to be visibly intoxicated while at appointments, but their drug tests were consistent, A, for the treatment they were supposed to be on, and then B, no additional positives. However, the providers were concerned that this was an in-house as well, that there was alcohol being distributed among patients, so they decided to add that ETG, ETS to their panel. This is another example of where cutoffs were important here. We talked through and made sure that the cutoffs were appropriate not to have positives for incidental use, such as non-alcoholic beverages. That ETG and ETS can be pretty, it can come up with even some incidental use at some larger volumes. The physicians were then trained for this addition and then specifically how positive results were going to be handled. I think that was a big part of the success with adding this to their panel, was making sure that all the physicians knew exactly the conversations they were going to be having with the patients and how they were going to be handling this as a practice with that addition and if there would be a positive. They also, as an aside, added breathalyzers to some of their stations. So in case there was a visibly intoxicated patient, they could do point of care testing. There was another pain management that really wanted to do an overhaul on their utilization. They went through an audit and they really wanted to kind of dig into their sub panels, and they weren't consistent across providers and wanted some info on whether their testing menu was appropriate. And I think this is a good, just a good exercise for a lot of practices in general, where they pulled their positivity rates on everything over the past year. And they looked at the most commonly positive items on their test panel and some that weren't. This was that example I talked about earlier, pulled that from this, where they found that for over a year, they hadn't had a positive for any synthetic cannabinoid or bath salt in their patient population, where they had had previously. And at that time, they decided that those were going to subsequently be removed from all of their subpanels, as they didn't see the clinical relevance to keep those in their panels any longer. And the last one I'll just touch on is a pain management practice that was looking into methamphetamine isomer. They had several patients, especially during cold and flu season, where they would be positive for methamphetamine and claim this was from an over-the-counter use of Vicks inhaler. I think there's a lot of things that you can Google out there and say, what could cause a positive? And they felt they really needed to differentiate and have a true report that said which isomer it was, whether that D or that L. So they did look through possibly adding that to their in-house lab, but the rates were so low, they came up with a send-out policy for that. So that particular, they decided not to add it to their in-house lab panel, but in these cases, they were going to develop a send-out policy for those. So it might not always make sense to add it to an in-house lab panel, but it might make sense to send those particular more rare types of testing out. So I have sources. As the slides go out, you'll be able to kind of look at those. But now I just really want to open the floor up for some Q&A. I know that sometimes that can be the most fruitful discussions. So looking forward to hearing your questions and any feedback you may have.
Thank you so much, Sarah. While we wait for any Q&As, I'm going to push the pop-up offer for people to register to our next webinar in this series. So the topic will be coding and billing for toxicology, and that will happen on December 13th. Feel free to click the link and register. Okay, I see the first question in the chat.
Kombucha. It's typically less than 1%, but there have been studies that show that it's not always very accurate reporting on those bottles. And it can actually go up as high as 3% in some cases, and it's inaccurate on the bottles. So there has been studies that have shown that kombucha can, if you drink a couple bottles, lead to positive ETS and ETG. So that's something that we sometimes do an FAQ about. A 6-pack of non-alcoholic beer could cause a positive ETS, ETG, but kombucha in general has some, especially there's a lot of local spots making it now. The accuracy of that reporting of the alcohol on the bottle has been shown to be very variable.
Thank you. And I saw another question before this webinar. Person, doctor asked me to ask you. It says, how often he or she should consult any physician with toxicologist or other laboratory expert for drug testing in their practice? So how often?
So how often should you reach out on reports or as general on like curating a test panel?
I think in general, maybe to review the current test panel if you have any, or if you need to add anything. So in general.
Yeah, I think biannually is a good time to look through and look at a test panel, unless there is a large drug of abuse or some type of epidemic or crisis that is coming on the market with a certain medication. But I think biannually is what I've seen most often practices kind of implement.
Okay. Wonderful. Thank you. Is there any? Okay, another.
Yeah, non-alcoholic beverages, NA, yep. A lot of time they're de-alkalized and they don't remove all of it. I think it's less than 0.5% is considered an NA beverage, but often, like I mentioned, there have been studies that even non-alcoholic beers around a 6-pack could lead to a positive ETS, ETG. Definitely low levels. That's why some people, a lot of labs have increased some of those cutoffs to make sure that incidental use isn't flagged.
And I have one more question.
Okay.
So there was about challenges in test panel customization. The question was, what would usually is the biggest challenge for doctors in customizing a drug test panel for patients?
I think the biggest challenge is trying to meet with that medical necessity and fall into that proper utilization. As I talked about those CPT codes and 1 through 7 and 7 through 14, when you start adding metabolites, you can easily fill up a panel pretty quickly. So really honing in on that patient population, what you're going to be prescribing and what's testing positive, and kind of moving away from some of the historical compounds that might be on a test menu but never pop positive. I think that's a hard switch to make. A lot of times those medications that are still drugs of abuse, still concerns, LSD, PCP, they may very well be positive on a rare occasion, but that might not be the current concern now. And kind of moving away from some of the older test menus to a new test menu is sometimes a challenging hurdle to overcome.
Robin, do you have any questions or maybe additions at the end?
If you have any other questions, my email is on the slides that will go out. Always feel free to email me, and fruitful discussion if there's any questions or just topics you want to chat through. I'm always enjoying talking through problems you may face as a practitioner and how the lab can be of assistance.
Wonderful. I enjoyed it, and I appreciate your time tonight. I know everybody's really starting to think about the holiday season and everything, but do consider joining us on December 13 as well. We will do the 4th in the series, and we will wrap up this fantastic series for CME. And I just wish everybody happy Thanksgiving.
Happy Thanksgiving to you as well.
Thank you. Good night, everybody.
Good night.